Today, I'm going to speak about a couple studies, primarily from the Rakai community cohort study, and the molecular data associated with those couple studies that helped inform how HIV transmits and the damage it does in a population. And it's really a celebration of how the epidemiological data and the molecular data intertwine, and how you can, how it's really the combination of both, which gives you so much information. So the goals for today, why can't I? There we go. Are to discuss viral factors associated with transmission. Anything to do with transmission is highly dependent on linkage. And what does linkage mean? So how do you establish that? Viral factors associated with pathogenesis, and then the combination of those for how that affects population level changes, both in HIV subtype distribution, but also what are potential, like how is the virus moving through that population? And that has to do also with determining the directionality of transmission and how that overlays on spatial studies, and how the combination of all of those really helps you understand how the virus is moving through a population. And the point to all of this is this gives us the targets for intervention. It gives us the ability to understand, are we actually controlling the epidemic? Are we controlling, are we reducing the numbers of new infections and the impact of that, of the HIV epidemic? So Rakai, Uganda is located in southern Uganda. It borders Lake Victoria and also Tanzania. It was the epicenter of what was first discovered as, or described as, slim disease in East Africa. It is still one of the areas with the highest burden of HIV disease in Uganda. It's got a prevalence of about 15%. And incidence, I guess that's debatable, but probably about 0.5% per year. The Rakai Community Cohort Study is a population-based study. It's based on, like 50 communities or so. There have been thousands upon thousands upon thousands of study participants since it was first implemented in 1994. There have been 20 survey rounds. The surveys were based on actually which particular houses. And so for these couple studies, that's based a lot on retrospective analysis of the data. Now, ART was introduced in Rakai, Uganda in 2004 with PEPFAR. There was also scale up, really, that and the male circumcision trial, which happened after 2006. And the census data of the individuals within the cohort has an unbelievably rich amount of data, both from the household census, sociodemographic and behavioral data. And it's really that data that the individual participants trust the researchers with that then gets merged with the molecular data that provides such a rich understanding of viral infection and transmission and pathogenesis within this population. So The impact of viral factors on HIV transmission and linkage. So you have to establish, did person A infect person B, to really make to understand, you know, what affects that viral transmission. If you can't establish that linkage, then really a lot of these studies can't be done. But among partnerships in Rakai, Uganda, the first paper describing the impact of viral load on transmission was headed by my boss, Tom Quinn. This was published in 2000. And one of the key features here is demonstrating that if your partner did not have a set point viral load above 400 copies per mil, no transmission was seen. Also, one of the sort of the asides that was presented in this paper was that if the male partner was circumcised, there was also a pretty substantial impact to not acquiring HIV. On this particular data set, later, Maria Wawer and Ron Bray, were part of the PIs of the Rakai cohort, also demonstrated that the stage of infection was critical to transmission. So when a person was in their early stage of infection, really acute infection, you were much more likely to transmit to your partner than you were during the prevalent stage of infection. For this particular paper, I was tasked with actually providing linkage. And that led to me getting more and more confused about what does linkage mean. Now, one of the great things about this particular cohort is that you have so much information, and you have couples that claim to be in a monogamous relationship and couples that do not claim to be in a monogamous relationship. And we found that there was that far greater linkage, and in terms of what was considered linkage at that time, which was a genetic distance of less than 1.5% and an 80% bootstrap support, that monogamy essentially could be validated molecularly, but that it wasn't cut and dry in terms of what was linked. And there was actually a bit of confusion, but from that point on, we used monogamous couples to determine, what are factors associated with transmission. We limited it to couples who claim to be in a monogamous relationship. And in one of those studies, led by Noah Kiwanuka, it was looking at, okay, what are viral factors associated with the transmission of HIV? And clearly, the greater the amount of virus that your partner had a substantial increase in risk of you getting infected. And so if your set point was above 75,000 copies per mil, relative to below 3,200, you're 14 times more likely to get infected. Also, the incidence or prevalent state of infection, very much so. Also, having your partner being younger. But what was also surprising was that subtype was associated with higher risk of infection. If your partner was infected with subtype A, you were 4 times more likely to get infected than if your partner was infected with subtype D. And so this led us to other studies of what are these viral factors associated with transmission. At a CROI presentation, Christoph Fraser proposed that HIV's viral load was a heritable trait, which at the time was was pretty controversial. And I came to him after his presentation and I said, I think I have data which may help either prove or disprove your theory. And then Deirdre Hollingsworth and Christoph did a lot of work in terms of determining whether or not this was a heritable trait. And they determined that, yes, about a third of the variation in set point viral load could be attributed to the set point viral load of the person who infected you. So now you've got a virus which the viral load is certainly predictive of transmission. Subtype is predictive of transmission. And then there's something within the virus itself in terms of the genetic information within there, which is also predictive, which predicts how much virus you would have. So we're still hemming and hawing about like, okay, what is a linked transmission? But I think there's getting more information on that. And frankly, 90% of that is pretty straightforward. It's the last 10% which seems to be getting a lot of people, a lot of debate. Viral load is certainly the primary predictor of HIV transmission. The stage of infection is associated with differential risk of transmission. Viral load's a heritable trait, and subtype A seems to be more transmissible than subtype D. When we switch to looking at pathogenesis, this is a description of the interaction between viral load and loss of CD4 count, which is actually a figure which was generated by Wikipedia. They actually have a graphics department. And so here what we see is the viral load, which is in red, which initially after infection, it rapidly increases. And then within a month or so, then decreases down to what is called the set point viral load. That is the relatively flat amount of virus, which can differentiate anywhere from 10 to the really almost undetectable levels to 1 million copies per mil. That amount directly influences the slope of the blue line, which is how quickly you are losing your CD4 counts. So the CD4 counts here are in blue, and you see that initial deflection of CD4 count with initial infection, and then it bounces back up when the viral load is contained. And then that slope going downward to AIDS and death is directly related to how high that set point viral load is. Now, when we looked at the infecting subtype and what the difference in set point viral load was among all these individuals in Rakai, we noticed that there really wasn't a difference in the set point viral load by subtype. But when we look at the Kaplan-Meier survival curves here, with red being subtype D and blue being subtype A, you see a very profound difference in survival between subtype A and subtype D. Now, in this graph, we have stratified by set point viral load, either above or below 40,000 copies per mil. And you see that the curves are basically offset. And so you have the impact of viral load is actually very stable within both of those subtypes. but within this, but between the subtypes, you actually have a very different impact of disease progression, where being infected with subtype D is almost like having an extra log and a half higher set point viral load than subtype A. So what's the difference? What makes disease progression so much faster in subtype D? Well, one of the first things we thought about was receptor usage. And what we found of these samples, we sent them to Monogram for co-receptor usage testing. And subtype D, almost a fifth of those samples of individuals within their first year of infection were either X4 or dual tropic, which was much higher than we saw in subtype A or recombinant strains of the virus. And among those individuals who had X4 tropic virus within the first year of infection, their median time to death was less than 5 years. It was incredibly high rate of disease progression. So certainly this X4 tropic virus was associated with higher rates of disease progression. So then Untek from who was a postdoctoral fellow in our lab, we had all this data. Now, this was the time way before full-length sequencing and all the beautiful types of data we have today. This was actually a time where we had probe data for 5 different regions of the virus, and that's how we established what subtype you were infected with. And so he looked independently at all those individuals where we had known dates of seroconversion and disease outcome, and looked for, okay, among these individual regions, what was most associated with rapid disease progression, or differential disease progression between subtype A and subtype D. And much to our surprise, it was a polymerase chain that seemed to have the greatest predictive impact of disease progression. And so again, we went to Monogram, and again, we sent our samples, and this time they performed replication capacity testing. And we found that subtype D had much greater replication capacity than subtype A. One thing which I've always found very confusing about that particular assay, and maybe it's a misnomer, but certainly has to be an interaction between replication capacity and how much virus you have in your blood. But replication capacity has no correlation with set point viral load. That is the graphic here on the bottom right, where you see replication capacity for the subtype B samples in red, and for the subtype A samples in blue, and what those set point viral loads are in log scale. And yes, there is no correlation with the replication capacity and set point viral load. There is a difference, a great difference in terms of subtype and replication capacity. And when Untek looked at, for subtype D, stratifying by high and low replication capacity, those with the highest replication capacity had a significantly higher rate of disease progression than those with lower replication capacity. The difference in replication capacity by subtype was also validated on Monogram's internal cohorts with subtype A and D. So that seems to be consistent across subtype. Let's see. So if you don't mind, I'll get to your questions at the end of this presentation. But no, we don't have HLA data on these individuals, unfortunately. Yeah, I wish I knew how replication capacity was not associated with disease progression. So, Oh, wait a second. Now I'm contradicting myself. All right. I will continue this presentation. Let's see. All right. So the summary of qualitative differences that impact HIV pathogenesis. Subtype D is more pathogenic than A. D expresses more exotropic virus than A. Exotropic virus is associated with rapid disease progression. And higher replication capacity is seen in subtype DNA, and that replication capacity seems to be independent of viral load as predictive of disease progression. Now, so in Rakai, we have subtype A and subtype D, and A seems to be more infectious, and D seems to be more A seems to be more infectious, and D seems to be more pathogenic. So if that's true, then on a population level, we should see subtype A replace subtype D. And to that effect, I asked one of the young people, young postbac ERDAs in our laboratory, you, Alicia, get to sequence everybody from Rakai from 1994 and 95. And what she found there was that 70% of those individuals were infected with subtype D. And this was also based on just fragments of GP41 and GAG. So we had a 5' and 3' end of the virus. And if you were consistent in terms of being subtype D for both, we'd consider you subtype D infected. If they were discordant, then you would be recombinant. Again, not the type of data we have today, just much simpler. So in Alicia's study, we found, yes, that subtype D was most prevalent strain. We also saw there seemed to be a gradient of more subtype D closer to the southern portion of the Rakai region than the northern portion. So there seemed to be a geographic stratification, well, not really, sort of gradient of viral distribution. The next postbac ERDA in the lab, Samantha Conroy, I said, guess what? You get to sequence everybody from the cohort in 2002, because we should see this difference in subtype distribution if really subtype A is more transmissible and D is more pathogenic. And that's what happened. We now subtype D had decreased in prevalence by 20%, and subtype A had increased. And when we looked at the differences by age group, we noticed that the greatest difference was actually seen in the youngest people, which does substantiate that subtype A is more transmissible than subtype B. We've had some more survey rounds sequenced, and one of them by Kate, who then turned it into a much more exciting study. But we looked at 6 regions of the Rakai community cohort, which have been steadily sequenced and surveyed over time. And you can see in those particular regions. Now the regions, the map here shows where those particular regions are. So you see region 1 is really quite central there. Region 1 next to region 9. But for these, again, subtype D prevalence is in, or should I say, the proportion of all the viral sequences that we have Subtype D, that percentage is in red. Subtype A is in blue. The recombinant strains are in purple, and subtype C is in green. And you can see for the regions over time, the first column is 94, 95, then 2002, 2003, 2008, 2009, 2011, 2012. And you see that steady decrease in the proportion of those viral variants, which are subtype D, with the decrease in the red lines. The increase in the blue lines would be the increase in the proportion of subtype A. And then you also see a fairly steady increase in the purple bars, which would be the recombinant viruses. What's also key here is that we have seen subtype C, but it really is not taking off. So Kate, we lured her to our lab and said, why don't you sequence everybody from 2009? And then she did just so much more, just really awesome work. And so one of the things she did was to link up all the epi data and the geographic data, and showed that, because the question was really about, within one of those communities, is there more likely to be linked? Are you going to see an epidemiological, a virological gradient of linkage, which is more concentrated within a particular geographic area? And really that did not exist. So you have the greatest linkage within a household, but once you get past the household, the distances to the within a community versus a region versus essentially the whole cohort, there is no difference. There is no signal. So really you have a lot of household transmission, but beyond the household, there really isn't any type of signal. And so that actually has pretty profound implications on treatment as prevention if you're trying to saturate a particular location with antiretroviral drugs. And if you're going beyond the house, once you're really not going to get that big of an impact beyond the couples, because so many of your new infections are not coming from within that community. So in terms of this population level changes, we saw subtype D decreasing, recombinant strains in subtype A increasing. 40% of the transmissions that were seen, according to Kate's study, was really within the household partnerships. But once you got outside the partnerships, most of them came from outside the community. Now, with the advent of deep sequencing, directionality of transmission actually became a viable area of research at really a population level. Before deep sequencing, you would need to do either limit of dilution PCR and sequencing, or cloning of those products and sequencing, both of which are incredibly labor intensive. So it would be very difficult to do at scale. But the directionality of transmission is really based on two bits of data. One is that the molecular clock of HIV, and really this goes for a lot of viruses, but that there's a relationship with viral divergence with time. And so that, and this was here. I love this picture of Thomas Leitner. He looks so stern. Anyway, so I believe what this man is telling me. That's but that as you have multiple sequences from multiple time points, they get further from that initial sampling. And that when you have, so that the distance in the tree is related to time. But the rate of those distances can actually be quite varied by individual. There's also, this is more work by his group with Ethan Romano Servzen. I don't know if I'm pronouncing his name right, but that really the structure of the phylogenetic tree that you have can demonstrate directionality of transmission. And so if you look here where the red sequences are from the individual who transmitted to the blue individual, and the shape of the tree, depending on how, would give you either consistent or inconsistent or equivocal outcomes, but that really with that temporal relationship, you would be able to see that the red sequences were, the blue sequences were derived from the red sequence. And so you have monophyletic trees where in that case, depending on what the difference in the rates are, you don't know if the red transmitted to blue. In paraphyletic, you actually have trees which would be very consistent, and also with what are called polyphyletic trees. Now, to help determine whether or not this theory is actually real, I went to the HPGN 052 trial. And in that trial, there were individuals who were placed either on immediate or delayed antiretroviral treatment, and their partners were monitored over time. And then when the partner became infected, there were sequences from the originally positive partner and then from the newly infected partner. And linkage studies were performed to determine, was the newly infected individual actually infected by the prevalent positive partner. One of the things which I love about this was that that data was actually sent to a number of experts and voted on whether or not linkage occurred, which again underscores that how not cut and dry linkage is. So within the primary trial, there were 11 out of 39 couples in which the originally negative partner got infected, were determined not to come from their prevalent positive partner. Now we were able to generate next generation sequence data on these couples, and actually on more than 52 couples by the time we had these samples. And this was really led by Rebecca Rose, who I asked to look at this data with me. And this is a rather complicated graphic, which then got summarized in a much simpler way for the paper. But so we had 52 potential pairs. Then we had to look at okay, how many different types of sequences did we have in terms of different time points for each potential transmission pair? How many of those pairs had evidence of linkage. And so, what was it, 14 of the 52 were demonstrated not to be linked. Then you also have the issue of multiplicity of infection, which can be problematic in terms of being able to establish linkage. Then, all right, in terms of looking at the trees and the topology of those trees among those linked pairs and determining whether or not, because we knew the direction of the transmission, whether or not the phylogenetic trees demonstrated that had happened. And so here, this distilled down to, again, fewer pairs, because we're looking at pairs which didn't have evidence of mixed infection. And based on this data, we showed that we had, and this is 454 sequencing of, so that's a particular, and of the GP41 fragment. So this is just a very divergent or diverse fragment of HIV, showed that we could predict correctly about 70% of the time. This helped, hopefully, I'm thinking it helped influence what was done in Pangea. And I think Pangea is just absolutely fantastic in terms of being able to get these full-length, deep sequencing data from all these different cohorts to really better understand, again, how HIV is moving through a population and what factors of the virus influence both transmission and pathogenesis. And so this was wonderful work done by Chris Wyman in terms of phyloscanner being able to take all that deep sequencing data and build the phylogenetic trees as you go through the entire genome. And so based on that data, and also work from Ollie Ratman and, again, Kate Kabrowski, because with the Rakai epi data, we know the timing of the infection. And so here you can, again, see to validate does your molecular data support what was seen in the actual population. And so in this case, using this particular technology on much bigger sequence data set, you could accurately determine the true direction of transmission at about 84%. So this gives us some amazing tools to then look in the population. And so there was this thought about treating these hotspots, that you have these areas where you have high prevalence of infection, high incidence of infection, presumably, and do those areas actually seed the infection in the lower prevalence areas? So this was work done by Larry Chang and Kate Kabrowski. And you have, again, here's the Rakai region, and you're using data from the cohort, and you've got areas along the coast of Lake Victoria, which are of high prevalence. And are those areas of high prevalence seeding the infections in the low prevalence area? And so you've got a lot lower, there's a lot fewer people who live along the coast than in the central farming regions, but the prevalence is so much higher along the coast. And so this was work done by Kate and Ollie Ratman, and I need to update that citation. But what they did was, with the sequence data and the location, they could determine these source and recipient pairs and they could see for those source and recipient pairs, what is the most likely direction of the transmission geographically. And what they saw was that there was far more transmission both within the fishing village and within the farming villages than really between the two. And that when you're looking at farming to fishing and fishing to farming, you're actually getting much more of the transmission going from the inland communities here marked in green than from the fishing communities here in red going to green. So going from green to red is much higher than 3 times higher than going from red to green. So that initial hypothesis of are the hot spots, at least in the Rakai district of the fishing villages, seeding the infections in the inland communities, turns out not to be the case. This type of use of geographic, epidemiological, and molecular data are also now being used in other population surveys, both in the Yasi communities in Botswana, also HPTN 071 in Zambia. And I really look forward to seeing presentations on from those two cohorts. So in terms of conclusions on these types of epidemiological studies on transmission and spatial studies, we see that the direction of HIV transmission can be inferred from sequence data, but it is not perfect. It is nowhere near perfect. But certainly it's very useful at a population level. And that hotspots are not necessarily the main source of new infections in the neighboring lower prevalence areas. And so targeting hotspots may not benefit the lower prevalence areas directly. So what I hope that I was able to show you is just how important it is to be able to link the epidemiological, geographical, and sequence data together. And also how there are different factors within the virus, within HIV, that impact both transmission and pathogenesis within the human host. And you can use these to really help understand how the virus is moving through the population. So there are so many people to thank. Particularly Kate Grabowski, who was a PhD student with me, who I felt I managed to lure to our lab. And then she did so many great, amazing things afterwards. Rebecca Rose, who really does so much great work with me. Certainly Lucy, who helped look at these slides before I presented them to you, making sure I wasn't going to lose people completely. Tom, who is always let me pursue research I'm interested in. So thank you.